Welcome to the like this talk where I will be talking about one of my PhD projects under supervision of Dr. Lindy Wall. Um, so so as you know, when DNA gets replicated, like different types of mutations can happen, like substitutions, insertions, deletions, duplications, etc. But what I'm interested in is like the classification within single base substitutions. So the basic, like one of the basic kind of distinctions between the types of single base substitutions is the transition transversions distinction. And so for like in any organism, you can like count how many of these two you have and then find the frequency of the sample, how each one of them is getting sampled. And that gives you the spectrum of like these two. But you can go even deeper to a deeper level and consider a more detailed level, which is the 1-mer level, in which you like an A to G mutation is different from a G to A mutation. They're not in the same class, and so you have more types of mutation to work with. And you can even go deeper to the 3-mer level, where you consider the context into account. So for instance, a C to A mutation in this level would be different if the C comes between an A and a G, compared to when it comes between an A and a C and so on. So these are different types of mutations when you consider the context into account. You can consider bigger contexts, but these are the 3 levels that I worked on in this project. There were enough to find the results. So let me just define what a bias spectrum is. So if you have, let's say this is a toy example. So if you have 6 types of mutations, they can be any 6 types of mutations. And then you count like the observed mutations in an organism, you might see something like this. So 2 types of mutations are overrepresented compared to these rare mutations. So these are underrepresented. So this is a kind of a biased spectrum. If it wasn't unbiased, then all of them will be equally likely. But most organisms are actually in nature biased towards one type of mutation or the other. And these rare mutations can have like some beneficial mutations that are harder to access, the organism, they are harder for the organism to access, given this bias. But these rare mutations can be accessed by different mechanisms. One way is to increase the overall mutation rate for all mutations. It will increase the access to these mutations, but this means that these will be accessed, and so this can be beneficial for an adaptive population. Another possibility is a shift in the bias, in the sense that we reverse the bias. So these two that were overrepresented can become underrepresented, and the ones that were rare can become overrepresented, and that also gives access to the mutations that were like that the organism did not have access to before. So in previous work, we worked on a theoretical like kind of framework to study the interaction between the changes in mutation rates and spectra, and the interaction between all of these possibilities. And this is the QR code if you're interested in. But the question of the current project that I'm presenting today is whether this applies to cancer or not. So as you know, cancer cells carry somatic cells generally like accumulate mutations, but if a mutation can lead to cancer, then that is called a cancer driver mutation. And these are generally suppressed and they're rare in normal cells, but they get sampled in cancer cells more. And so the question is whether these cancer driver mutations were accessed only because cancer has a higher mutation rate, or is there also a reversal in the bias in cancer cells as well that give access to these mutations? So we worked on the PEACOCK data set. We have mutational data from 20 different tissues. The samples can be classified into hypermutated cancer samples and non-hypermutated samples. So the hypermutated ones have very high mutation rate, more than 10 mutations per megabase, while the others, we just call them non-hypermutators. And this is distribution across tissues. I will be using these colors across like a few slides. Yellow will be for normal cells, evil faces will be for cancer cells, and then they will be blue for non-hypermutators and red for hypermutators. So the first thing we wanted to test whether there is a difference between the three-mer spectrum between the different tissues. So for instance, we want to test whether bladder has a different spectrum from the other hypermutated samples from the other tissues or not. So we just correlated the three-mer spectra for each individual tissue with the overall spectrum from the other tissues in the same category. And we found that within non-hypermutators, there's a very high correlation coefficient, suggesting that there's like a similarity between the spectra across tissues and across donors, but that is not correct in hypermutators. And so, I mean, our goal is to test the biases, but this suggests that if there's a kind of a reversal in the one of these tissues in non-hypermutated samples, then maybe there should be also a reversal in the others as well, because they're highly correlated. But this might not apply for hypermutators, of course, because of this lower correlations. So now we want to test the bias for each the biases and the changes in the biases. But the question is, what kind of spectrum are we comparing with? So if we want to test whether the cancer spectrum is reversing a bias, then what kind of bias are we comparing with? And the answer for that is the germline spectrum. So we consider germline is like the basic kind of cell, and it's like the first human cell that like all other cells come from. And so like any change in the bias later will be a reversing of this like prevailing bias. And so we are comparing the cancer spectrum with the germline spectrum. But the issue is that in this toy example, the reversal here is like perfect, because the oversampled types became undersampled in this spectrum, but the others, like the ones that were undersampled, now become, all of them are oversampled. But in reality, and especially when you have like 96 types of mutations, like in the three-mer level analysis, then you might not see this perfect change. So you might have a lower from this one, but more from this one. And so this is like a conflicting effect. And so we wanted a measure that gives us, that summarizes all of these effects across the different types. And so it simply adds the magnitude and takes the direction of the change into account and gives you a single number that represents whether you have a reversal or not. If that number, so we call that the bias reversal measure. If this number is positive, then there is a bias reversal, and that is, like, according to the hypothesis, that's good, which means like it gives access to the driver mutations. But if it's negative, then there's a bias reinforcement, and that's hypothetically bad. So these are the results. These are the distributions across different tissues for the bias reversal measure on the 3 levels of analysis: the transition, transvergent, the 1-mer level, and the 3-mer level. Again, in yellow, blue, and red for normal, non-hypermutators, and hypermutators. And you can see that hypermutators on all levels of analysis in all tissues have positive reversal measure, which means that, like, in all tissues, we have a reversal in the bias, which meets our hypothesis, but this does not apply to hypermutators or normal tissues, which suggests that maybe the mechanism in which the hypermutators got access to these driver mutations is simply because they have a higher mutation rate. So we also wanted to, like, test whether there is a kind of a biology behind this, like what kind of, so we have a spectrum that's very similar, that we kind of have a parallel evolution of a spectrum across all tissues and across all patients, and it reverses the bias. So we wanted to see if there's a biology behind this, like what kind of mechanism can reverse the bias and cause this. And so again, because these were very similar, we compacted them in one spectrum, called it the non-hypermutators spectrum, and performed the signature decomposition based on the cosmic single base substitution signatures. And so we found that about 50% of the mutations are attributed to signature 5, but this is only a germline mutational process. It's not very interesting. But what's interesting is that there's about 14% of the mutations attributed to signature 40, and that is very recently got attributed to hypoxia, which we know is very common in cancer. And this signature reverses the germline bias very strongly. And actually, most of the other signatures also reverse the germline bias that we found, but like this was the most interesting one, and I don't have time to discuss the others, but this was very interesting. So in summary, we have a bias reversal in non-hypermutators samples, but no such pattern exists in hypermutators. We have a similar spectra, kind of a parallel evolution happening in non-hypermutators, but again, different spectra in hypermutators, which suggests that cancer is spectrum-driven in non-hypermutators, but hypermutators access driver mutations probably only because they have high mutation rates. And these are the funders for this project. This is the QR code for our preprint. If you're interested in more, here's my email if you want to have any discussion, and I'm happy to take any questions.
Great, thank you so much, Marwa. A great talk. So we do have a couple of minutes for questions. Jigar, I will unmute or allow you to unmute, Jigar, and you can ask your question. Oh, oops, sorry, I just muted you again. Try. There you go.
Hi, Alex. Thanks. Hey, Marwa, great talk. I was just wondering if you saw any particular mutation type elevated, since you claim that in hypermutators the mutation rates are elevated in general. So was there a particular mutation type that you saw elevated, which was acting as a mutator and driving up the mutation rate?
Yeah, so the hypermutators are mostly in colon and skin. So we did do a signature decomposition for these for these two in specific, not for the others because we didn't have enough mutations. So in skin, most of the hypermutation comes from UV light exposure signatures. And that's like the kind of hypermutation happening in skin. In colon, it's like I don't remember the numbers of the signatures, but it's mainly mismatch repair, mismatch DNA repair mechanisms that are driving this high mutation rates.
All right, great.
Great, thank you so much. Okay, so thanks again, Marwa, for a very nice talk. And our next speaker,